CSIR Net Life Sciences. This exam is just around the corner and I believe all attendees are in the full fledged of their preparation. But still, you might need a few tips and tricks for how to select questions during the exam. You might think, how does it even matter? Why question selection matters in exam? Well, we know that for the CSIR exam questions, there is negative marking, which means 25% will be deducted for each wrong answer. CSIR net exam is more of a rejection game than a selection game because majority of candidates get rejected. So what you need to do if you want to get selected, you have to decide on the questions wisely so that you can avoid the negative markings. It's not that which questions you can. It's the questions which you can answer confidently, which you know that has least chances of negative marking. And also there is an art of scanning easy questions to be away from the trap questions or tempting questions which might have more chances of getting you negative marking. So let's dig into the video today. I'm sure this is going to help you in the upcoming exam. Decision making while selecting question is very important for this exam. Don't attempt questions that you are not very sure of. We know that in Part C questions, you have to attempt total 25 questions. Out of 75 questions, you have to attempt only 25, isn't it? But even if you attempt 50 questions from Part C and all of them you are getting it right, then each of the Part C questions contain 4 marks. Totally, you will be getting 60 marks. But if you attempt 20 questions, 15 out of which 15 questions you know are right, that's why you have answered correctly, but you also tried to answer the ones which you were not so much confident about, and you got them wrong. That means -5 will be deducted from here. Then your score comes to 55. You might think that if I answer more questions, I will have more chances of getting my marks increased. Well, that applies to a certain extent. You have to attend at least a minimum number of questions. I'm not saying that if you attend just 5 to 10 questions in Part C, rather than 15, your marks increasing chance will be more. No, not like that. After a certain limit, don't push yourself to answer more questions. That is what I'm trying to tell you here. So decision making on which questions to select, which questions to reject is an art. Okay, so it's like Jenga. You know, a wrong decision can actually cost you your calculation. So you have to choose the questions very carefully. It's all about planning, the risk and strategy. So what kind of planning? You have to know how much qualifying marks you need. For example, generally if you qualify, if you have 120 marks out of 200 in general category, and if you are attempting to qualify for JRF, then 120 marks with that you can easily qualify JRF exam. That means out of 200 you're looking for 120 marks. That is planning. Okay. Now in 120 marks in Part C, how much minimum marks you would need? We know that if you attend all the 25 questions right in Part C, then you will get 100 marks. And even if you're not able to attend 25, then at least if you get 20 into 4, that is 80 marks. Still you need 40 marks. So another 40 marks, 30 marks you can get from Part B questions. So 15 into 2 will be 30. And another 5 marks questions, 5 questions from Part A. So that will fetch you another 10. So this is how you have to do the planning. And then you have to take the risk. Okay. So 20 questions from Part C, 15 questions from Part B, and 5 questions from Part A. This is minimum what I'm saying, that you must attend to at least get this JRF in general category, provided you have to get all of this correct. From this, if you get negative marking, then again the strategy will not work. So let me show you examples of the types of question that you get in CSIR. One is direct question. Direct question means there is no twist in that. It's clear interpretation, and mostly these are from your memory, recollection of memory. Most chances are that with a decent preparation you will get them right. Okay, so since at Biotechnica we suggest that you study 7 to 8 units out of the 13 syllabus to have a most chance of qualifying with least preparation. So minimum 7 to 8 units you should prepare properly. So out of this direct questions you attend from the units which you have studied, mostly you will be able to attend them without any chance of making mistake. Now, tempting questions are the ones which have chance of making a mistake, hence can be ignored. Most options might appear somewhat easy. It might look easy, but there may be some twist over there. Okay, so you have to guess the other 1, 2 option. Either you will need some guessing, or the question might look easy, but it might actually have more chances of making mistake. I will show you some examples. And another type is trap question. So these are like hard questions, time taking, and also they will involve lot of numericals. The options will be confusing. Okay, so definitely there will be some catch, which in fact not one catch, there will be several steps at which you will have more chances of making mistakes. So you will have to watch every step in solving that question. You have to be very focused and be very concentrating on the question to have it correct. So try to avoid those questions. All you have to do is you have to select 20 part C and 15 part B questions. So select the questions mostly from direct, or I will give you some tips which type of questions you will select. Okay, first is the art of identifying easy questions. So here are the easy question example. As you can see, which one of the following is not an extracellular matrix protein? As I said, if you have done a decent preparation, this is from unit 4, anyone will say that keratin is present inside the cell. Okay, so it is a type of intermediate filament. It is a type of cytoskeleton, so it is present inside the cell. Then the energy rich fuel molecule produced in the TCA cycle. Everyone who has preparation of unit 1 will know about the TCA cycle, that 1 GTP, 3 NADH and 1 FADH2 is produced. So no mistake in that. These are direct questions. Learn the art of scanning easy question. Some questions might look lengthy, but again they are direct. Okay, so this art will come with more and more practice. Try to solve previous year questions, go through the full length question, and within a limited time frame, try to sort out or filter the question that you would want to answer, and see how your marks, the total marks, is adding up to. So at least you have to solve 10 question papers minimum, previous year questions, to have this art developed for the exam. Okay, like fluorescently tagged protein was used to study protein secretion in yeast. So fluorescence was observed in all these 3. So we all know that this is the part of the secretory pathway. So you have to tell from where all it will start, right? So most of you would be knowing that the protein is always synthesized in the rough ER. So obviously C will come first, and then only it will be going to the Golgi, because ER is connected with this Golgi, with this lamellae, and then it will be packed into the secretory vesicles and it will be sent out. Okay, so which of the following is a group of signaling molecules that act as morphogens during development, and its effects are mediated through the receptor patched and binding partner smoothened? Again, this is a very easy question from unit 5. If you have studied the signaling pathways of unit 5, everyone even in biotechnica classes, when we teach these signaling pathways, we make sure you know about the signaling molecule, which in this case will be hedgehog, the receptor molecule which is patched, and any other associated secondary molecule, which in this case is smoothened or commonly called as SMO. So the answer over here will be hedgehog protein, right? Some more direct question examples. Now here it is a direct question. You should learn what is the difference between direct and easy. So this is a direct question, like pH 4 and pH 10 are mixed. What will be the approximate pH of the final solution? But it is not an easy question. Okay, so so together it doesn't come to like average of this 4 and 10 divided by 2. It's 7, so it's not like that. There is a different catch for this. So learn like only if you know such kind of questions, then only attend. Don't attend if you are not sure of that. Okay, another, which one is required for vitamin B12 absorption in small intestine? This is, if you are not knowing the question, if you're not knowing the answer, it's better not to make a guess. So these are direct questions, but not necessarily easy questions. Okay, only if you have studied that, then only you should attend those questions. Next are tempting questions. Do not fall into the temptation. One example of tempting question, as you can see, some errors occur during DNA replication that are not corrected by proofreading activity of DNA polymerase. So defect in the activities of some of the following enzymes impair that process. Which combination is correct? This question appears tempting as all of the names given in the options, they are part of the DNA repair mechanisms. So we might end up selecting all three combinations given in A, B, C, right? But here we need to carefully look at that part of the question which is saying errors which are not corrected by proofreading activity of DNA polymerase. That indicates we need to look for a repair system which works immediately during or after replication. And that one is mismatch repair, where only enzymes given in A and C are applicable. So the correct answer over here is 4. That's why you need to be careful. Look at another tempting question. There can't be any better example than this. The word fermentation is used in biochemistry and microbial technology to denote different phenomena. Now, the fermentation, they have used the letter C, and microbial technology, they have used the letter T. So this is where instead of writing fermentation and microbial technology, they have played with the words C and T. So if directly they had asked like all fermentation is microbial technology and all microbial technology is not fermentation, reading that would have been easy, but instead they have said like that. So be careful with this kind of questions. Okay, there might be chance of making mistake. Beware of the trap questions. Now, what a trap question? Most chances that you will get confused. One example, this is regarding from unit 13. Now, to answer this kind of question, as you can see here, many words are used like polymorphic, then transcription start site, then the heterozygous. So all these things, heterozygous, transcription site, restriction, digestion, you have to know all these concepts. You have to be familiar with this. Students often fail to understand such type of questions as it is complicated. And there are chances that in solving this question at different steps, there are chances of making mistakes. So you have to cross all those steps properly without making mistake. Then only you can come to the final answer. Okay. Another trap question example is not a numerical problem, but in this case, first of all, each of the sentences, each of the statements are from different biomolecules. This is from unit 1, but as you can see, this is from carbohydrates, first 2, then the third is from amino acid, fourth is from nucleic acid, fifth is from fatty acid. So there are chances that you may not have studied one of the biomolecule. So such kind of combined questions you are supposed to avoid. Choose the option that represents the combination of all the correct statements. Now options also, if you look, one is having all 4 options, then 3, and some is having only 2 options. Try to answer questions which have easier combination of option statements. Like they will ask A and C or B and D or A and D. So those are easier, but these have more chances of making mistake. So better to avoid such questions. Another trap question example, as you can see, this question is based on the life table. Here you need to know, recall and apply all the formulas: mortality, age survivorship, fecundity, and then only net reproductive rate can be calculated. So unless you are not very sure, try not to attempt this kind of question, as these are very lengthy also, and moreover filling one blank with wrong figure. Okay, if one of the figure values you are getting it wrong, it will lead you to the complete wrong answer and it will utilize your time also, leaving full chances for negative marking. So stay away from lengthy, time-taking, risk-involved questions. Remember your planning and your target. You only have to find out 20 part C questions and 15 part B questions and 5 part A. So select the question accordingly. Okay. When you go to the supermarket, what do? You pick up things which are easy to consume and which are cost effective. So in exam also you apply the same thing. Target more match the following and graph based questions. So these are easy to attain and these are very scoring topics as well. Okay, this will be not taking much time. Even if you know 1 or 2 matches, then you can come to the correct answer. Whereas you have to avoid the related combination unit questions. So 2 units together some question, or combination multiple subunits together in one question, unless you are very sure of the answer, or it is such kind of question is a graph based question or a match the following which is comparatively easier, then you can attend, else you try to avoid such kind of questions. Okay. Another trick is questions having opposing statements generally and most of the time, one of them will be correct, other will be wrong. Okay, so sometimes they can be easy if the statement is relatively simple, like reaction can take place spontaneously if delta G is negative or if delta G is positive. Obviously both cannot be correct, so one of them has to be correct, isn't it? Delta G provides no information, delta G provides information about rate of reaction. Again, opposing statements. And if you see here, option combinations are simple, A and C or B and C, so only two you have to eliminate. So in such kind of questions, elimination trick can easily land you to the correct answer. Okay. Then guessing can work in some questions, like when I was talking about easy and direct questions. This will be more helpful in part B, because the direct and small questions, easy questions, are generally from part B. So addition of the antibiotic, here cephalin is an inhibitor of what? It is saying it is leading to filamentation of the cell followed by lysis. Now lysis means something to do with the cell membrane or the cell wall. So peptidoglycan synthesis. This way you can guess. Okay, so this you will only do when you are falling short of the total number of questions that you need to attend. Let's say you have not come up to the 15 part B questions. Okay, then you can try to take this risk. As I said, question selection is a matter of risk. But guessing doesn't work in all questions, so be careful. Like in a signaling event, binding of an extracellular ligand GPCR, phospholipase B, which of the following represents function? So whether the phospholipase C makes these two or this to this or this to this two or this to this, if you don't know this thing, there is no way you can come to the right answer just by guessing. So don't take that risk. Okay, here also this is from unit 6, the light reaction. In that light reaction, the photosystem, they have asked the sequence of the electrons, how they are passed. Again, if you don't recollect it, probably you have studied the photosystem but don't know the sequence, then don't try to guess by imaginative figure, like a rough figure or a very blurry figure that you have in your mind. You try to figure out that, okay, I saw plastoquinone at the end, so let me guess. Like that, don't take the risk. Okay. Then a part C question can also be a very easy question, especially when it is either match the following or even graph based questions. Okay. For example, just see this unit 4. These are match the following. Let's say even if you have not studied this part, okay, but still you can see lectin, right? So selectin is there, so you can at least try to see if you are able to match. Again, spend time on such kind of questions which you have not studied. That unit, if you have not studied, then you can try looking at the options only if you fall short of your total number of questions that you're supposed to attend. Okay, because individually looking at it will take some time, right? Then cadherin, they are calcium dependent, so you can see mediates calcium dependent. With this only you can come to the right answer even if you have not studied it. Okay, same even in unit 5 also. Little bit if you know about the holoblastic and meroblastic cleavage, you can come to the right answer. And don't get fooled by the length of the question. Okay, so if somewhere you have studied that protein folding, when the protein is folded, it is a stable molecule, isn't it? A folded protein is a stable molecule and when any molecule is stable, when the energy is less. So as you can see, this is a graph where the energy is high at this point and energy is low at this point, which means that at this point it must be the folded protein. So D should be the native structure. So like that you can easily match the native structure with the D part, isn't it? So like that you may come to the right answer. It's not that only part B questions can be easy. Every paper you will find at least 4 to 5 such questions which are just visual analysis and requires no prior specific knowledge. And yeah, so A will be like where the protein is completely unfolded. That means structure with highest entropy. So with this two only you can come to the right answer. Okay. So yeah, combination questions, as I told you, try to avoid. For example, this is a combination question from unit 6 and unit 8. In order to answer this question correctly, you need a preparation of both this unit. If you have not studied any one of them, then try to avoid. Okay. So you are the decision maker. Remember these small tips. Move on if stuck. It is not a prestige issue that you have to complete it. If it is taking more than 3 minutes for a part C question, more than 5 minutes for a part C question, and more than 3 minutes for a part B question, or rather more than 2 minutes, then you can skip it. Okay. Do not spend more time and take risks only when desperately needed. As I said, target the qualifying marks, and accordingly skipping unsure questions will help you avoid the negative mark. You don't have to attempt every question from your studied unit. This is a very important point. It is going to help. If you have prepared 7 to 8 units, it's not necessary that every question that have come from those units you have to attempt. Okay, you can skip it if you are feeling confused. And know when to stop. Control your greed, that one more question, another question, but at least solve the minimum. There is a thin balance of knowing when to stop. Okay, like too much greed, or at least the minimum that I need to solve. Again, practice more to understand more about it. So remember, taking right decision in the exam can help you score more even than the topper. That's all for today. Let me know in the comment section if you found this video helpful. Thank you everyone.